Now that we've talked about what bacterial transformation actually is, let's take a look at its benefits. Now, transformation allows bacteria to adapt, which is huge. Um, we think of adaptation and evolution as being something that's primarily done by sexually reproducing organisms because sexually reproducing organisms, as we saw first semester, will have genetic variation as a result of meiosis and a result of random fertilization. But if you are a bacterium and you reproduce asexually, you just split into two, right? So that should not really give you the chance to adapt. All of the different bacteria in a colony would be identical copies of each other. But if a bacterium can change its genome throughout its life by taking in, assimilating plasmid DNA, that, in essence, has the same effect as sexual reproduction. In fact, when you pair this with the fact that bacteria can reproduce extremely quickly, overnight, this actually makes them better at adapting than we are, right? Like it says here, thousands of times faster when we look at the evolution of bacteria versus the evolution of a large multicellular organism like us. So whereas we need to look at generation upon generation upon generation, hundreds of years of, say, humans to actually see the signs of evolution, we can see the signs of evolution in bacteria over just a couple of days. So this is something that's totally unique to bacteria, but makes them pretty awesome. Now, this is probably most beneficial to bacteria in modern times because it allows bacteria to contend with antibiotic medications. If you think about it, antibiotics have only been around for the past, 80 years or something like that, maybe a little less. And so that's a new thing that is very, very dangerous to bacteria. Because again, we're looking at this from the bacterial perspective. Obviously, I'm at school here today. We're looking at this from the bacterial perspective, their point of view. So antibiotic medicines are terrible things, right? They kill the bacteria. But sometimes a bacterium will have a mutation, a beneficial mutation, that allows it to survive when confronted with a certain antibiotic medication. Now, that bacterium can share that genetic information, share that mutation via a plasmid with other bacteria. It can share that antibiotic resistance gene. So now it's not just that bacterium and its progeny, right, the cells that come from it, will survive the antibiotic. Maybe all the other bacteria around it will also learn to survive that antibiotic because of transformation. Now, if those bacteria multiply, we as humans have a battle on our hands because we are going to try to develop antibiotic medicines to get rid of the bacteria, and bacteria are going to evolve to resist those antibiotic medications. Sometimes we refer to this as an arms war because it's actually sort of like this battle back and forth, us trying to stay ahead of them and their evolution trying to stay ahead of our developmental science. Now, I have just a couple of examples for you. The first one is MRSA. You may have heard of this before, MRSA. MRSA stands for methicillin-resistant Staphylococcus aureus. You can see a slide with MRSA up here. There would be these dark circles, little clumps of staphylococci. And Staph aureus is a very, very common infectious agent. Staph aureus is a very common infection. Most of you guys have probably had a minor staph infection before. Staph can cause primarily cutaneous infections or infections of the skin, and also food poisoning or a gastrointestinal, right, a stomach infection. Staph grows very commonly on the skin. Staph grows very commonly on foods as they begin to spoil. So if you eat something with a lot of staph growing on it, that could cause you to have food poisoning. You have a bout of vomiting, lasts about a day, and then hopefully you're better. Many of you have probably experienced that in your life, or what I'm betting is true, most of you have probably experienced like an infected hair follicle before, maybe somewhere where you shave or something like that. That is probably, it doesn't have to, but that is probably a very minor staph infection. Now, staph can also cause really serious internal infections of the body, deeper tissue infections, and also sepsis, which would be basically an infection of the bloodstream that affects the entire body. Now, those serious infections and sepsis are going to be much more likely in people who are immunocompromised, meaning their immune system doesn't work very well. So maybe somebody who's very old, somebody with another disease, maybe someone who's very young, but they'd be very uncommon. However, today we are starting to see the existence, and this has been going on for several years now, but the existence of certain strains of staph who resist the common antibiotics we would use. Penicillin or methicillin, which is a derivative of penicillin, is a very common, it's the oldest antibiotic. It's actually very effective on normal staph infections. But we are seeing strains of staph that resist those antibiotics. And when you see that, those staph resist all of the penicillin-derived antibiotics. So things like methicillin, things like the pink stuff, amoxicillin, there's one called dapsillin, anything with psyllin in the name will not harm these resistant bacteria. And these are becoming more and more common. Now, probably what happened was you've got some bacteria with a mutation that can resist it. You've also got people who don't take their entire prescription when it is prescribed to them. That allows more of the bacteria to survive, more of them to transform. You end up with more of these resistant bacteria on your hands. So first thing you can do is always take your prescriptions all the way through. Even if you start to feel better after 3 days, take all 5 days of that medicine if that's what the doctor prescribed for you. Now, when we look at staph, we've really got two different types when we talk about resistant staph. Let's start here on the right. Community-acquired MRSA would be a resistant staph that you pick up at school or at home just randomly. And so you get this hair follicle. Maybe it looks a little worse than normal, the infection, a little more red, a little more puffy, more inflamed, and it just doesn't seem to go away on its own. And so you go to the doctor, they give you a cream for it, maybe a pill to take, and it still doesn't go away. You've probably got a community-acquired staph. You would probably need a different medicine to get rid of it. This is still not crazy common, but some people this happens. Generally, it takes a while to clear up, but it's also not life-threatening or anything of the sort, and it usually stays as a cutaneous infection. This has happened to my brother twice, strangely enough, which wouldn't be common at all for someone to do this 2 times, but my brother has had 2 different places. He had one kind of on his underarm and one spot on his back where they identified it as a MRSA infection, and he had to have a little extra treatment. This picture on the screen is a pretty serious case. That this person has let this go, but a lot of times people describe this as looking like a spider bite, and people just think, oh, I've got a spider bite or whatever, and it'll go away. Well, you need to realize spider bites are really, really, really uncommon. Really uncommon. So anytime as a doctor you see a spider bite, you're like, I don't really think that's what that is. I actually had a student several years ago who wanted me to see a spider bite she had. I think it was my 2nd or 3rd year here at Mercy. And I looked at it right away, and I knew it wasn't a spider bite, and I told her she should go to the doctor, and she did, and she had, similar to this picture, it wasn't nearly this bad, but location-wise, right on her ankle, she had a follicle that had a MRSA infection, and so she had to go through a couple weeks of medicines to get rid of it. Now again, community-acquired isn't so scary. What is scary is hospital-acquired MRSA. Now realize, these are MRSA bacteria that are hanging out in a hospital. They've seen it all, right? These bacteria have seen every medicine you've got. They're surviving in the hospital. It's like the most dangerous place for bacteria to be. And so if these guys are surviving, they're bad dudes. So if you get a hospital-acquired infection, it's probably a really pathogenic bacterium. It's probably really resistant to medicines. And you're in the hospital, right? You're probably already sick. Your immune system is already kind of taxed. Or maybe you have an open wound or something like that. So hospital-acquired MRSA is a lot more serious. It's usually a lot harder to treat, and it's a lot more likely to be one of those deep infections that could actually become lethal. And this is what has scared people, are these hospital-acquired cases that actually ended up in death for some patients. Now, just as a side note, this would never need to be known for the AP exam, but those penicillin drugs don't work. You have to use really expensive, powerful alternatives. Right now, you get a cocktail called vancomycin and teicoplanin. which are these really advanced drugs. Only way to get those is intravenously, and usually you don't even go with a normal IV, you go with a PICC line, which stands for peripherally inserted central catheter. This is where they basically lace a little tube up your blood vessels, and they're going to drip that medicine right at your heart so that it pumps out to your whole body. So pretty serious deal if you get a really bad, we'll say hospital-acquired MRSA that is septic in your whole body. Now the other example I don't have as much to say about, they don't know as much about it either, and that's C. diff, which stands for Clostridium difficile, pictured up here. C. diff causes an intestinal infection. It's not super common. It's an intestinal infection that causes painful diarrhea and cramping of the abdomen and the stomach. But if it goes on long enough, it can cause a life-threatening inflammation of your colon. It's called ulcerative colitis. Ulcerative indicating that it could open up your colon, basically. Your colon could rupture and things. So you don't want to get that, obviously. Now, what's interesting is that C. diff infections, or antibiotic-resistant C. diff infections, tend to come around after a patient has been taking antibiotics for some other kind of infection. Because what happens is you take those antibiotics to get rid of, your pneumonia or whatever you have, and you've inadvertently killed off a lot of the normal bacteria that live in your gut with those antibiotics. Now, because of that, resistant C. diff survived that antibiotic, and now it's basically alone in your stomach. It can thrive, it can multiply, there's not competition from other bacteria, and it will produce great amounts of toxins that cause that inflammation and that issue. If you just have some C. diff mixed in your gut with a bunch of other bacteria, there's usually enough competition between the bacteria that C. diff won't become an issue. Many of you potentially have actually been asked by your doctor to do something that helps resist a C. diff problem, and that is to take a probiotic whenever you go on an antibiotic. Maybe you haven't had that happen. I've had my doctor do that to me once. About a year ago, I got an antibiotic for a sinus infection, and then he also said, go ahead and take a probiotic as well, and take that probiotic for a couple weeks after you finish your antibiotic to help replenish that gut bacteria so that you don't leave any really bad guys in there, which is what the C. diff would be. Now, there is a new strain that was identified probably about a year and a half ago now that shows really strong antibiotic resistance and generates way more toxins than previous kinds, and is leading to lethal infections in people. Now, the last thing I noted here at the bottom is really kind of weird and creepy and has not hit in the United States yet, but overseas, particularly in some of the Asian countries, they are trying a treatment technique called a fecal transplant, which is exactly what it sounds like. They're taking feces from one person who is healthy and putting it into the colon of someone who has a C. diff infection. This is thought to, provide a whole bunch of normal bacteria back into that person that can compete with C. diff. It's weird and just sounds nasty to me, and if it involves handling poop, I don't really want anything to do with it. I don't know about you guys, but it can count me out. Now, from the human point of view, we also have the potential to manipulate bacterial transformation to help us some. Now, we saw a little bit of this in the first part of the biotechnology notes. We'll see a little bit more in the latter part of the biotechnology notes. But bacteria can be a vehicle for genetic engineering. We can tweak what genes are found in a plasmid, and then bacteria can carry that plasmid and can deposit that plasmid in cells, potentially with a gene that we want to get to those cells. Right? Bacteria will form recombinant DNA. Right? They will put DNA into other cells. So if we control what that DNA is, that could be really helpful. In fact, this is one way we could potentially, it's a big stretch, but start to kind of cure genetic diseases. If we could use bacteria to give people genes, that could fix their DNA maybe. Now, we're a long way from that, and viruses are actually probably a more logical way to do that, which we'll talk about later this year. But bacteria right now are used for amplifying genes in certain labs, as you see in the biotechnology notes. They're used a little bit for gene delivery, and they're primarily just used in research versus treatment of anybody else. Now, put this down here kind of as a joke, but us using and manipulating bacteria only works if we survive bacteria, right? And so if they continue to kind of out-evolve us and our medicines, we might have to worry a little more about infections than we maybe do today. Now, I put this book on here. Dr. Paul Ewald is a research scientist who actually works at the University of Louisville. He is internationally known for his actually somewhat controversial ideas about the evolution of diseases and the way we can manipulate that, and sort of new trends in medicine. And he's been asked to speak across the world at different conventions and things. And he has a really, really good TED Talk, if you've ever heard of TED Talks, which mirrors one of his lessons that I had with him when I had his class at the University of Louisville. So if any of you guys end up going to U of L, you can take a couple different classes with Dr. Ewald. I would strongly recommend it. And he's written several books, but this is the main book he wrote, called Plague Time, about kind of all about this, about the evolutionary ecology of disease, and the way that affects us as humans. I will have you guys watch that TED Talk potentially very soon, maybe even with an online snow day. But if not before, we'll also watch it when we hit evolution, because the great kind of intersection between this bacteriology stuff and evolution. And so it kind of talks about some things that we know right now, and it'll talk about some things that are coming right around the corner when we get to the evolution unit. Now, the last thing to talk about just momentarily, and we'll look at this in class as well on the day that we set up this lab, is our lab, the pGlo Plasmid. Now, the pGlo Plasmid is actually produced by a laboratory, right? We just order that from a biological company. It's not a naturally occurring plasmid. But the plasmid that we're going to use in our transformation has several genes on it. And now, I think this will make sense to you. So here's a picture of the pGlo Plasmid. You see all kinds of stuff going on here, but we'll break this down into simpler terms. On this plasmid, you are going to see a gene that should give the bacteria resistance to ampicillin, one of those penicillin-derived antibiotics. So that if we put the bacteria into an environment where we find ampicillin, those bacteria should not be able to survive. Now, the second gene that we put on here is the green fluorescent protein gene. Now, this is part of why this isn't a naturally occurring plasmid. Green fluorescent protein really comes from jellyfish more than anywhere else, and it should make the bacteria glow when we hit them with UV light. So if we give them these genes, right, they should be resistant to the ampicillin drug, and they should glow under UV lighting. Now, we also put on there something that we just learned about recently, an operon mechanism. Right, and this operon mechanism is the Ara-C operon, or the arabinose operon. What this does is when there is arabinose, a particular type of sugar, in the environment, bacteria will turn on expression of these ampicillin resistance and green fluorescent protein genes. If there's not arabinose in the environment, we won't turn on those genes. So in other words, you got to have the right sugar to serve as the on switch for resistance and for green fluorescence. So in our lab, we're going to attempt to transform some bacteria and see if we can get these genes to express, and then talk about how we can actually visualize that expression. So thanks for listening, and I will see you in class.